Hello everybody, so today we are going to continue our series about the macromolecules and we are going to talk about the nucleic acids. So what are the nucleic acids? Nucleic acids are the macromolecules that store and transmit hereditary information. This definition might sound abstract, but let's look at their building blocks. They are just made out from the nucleotides, which are made up from the phosphate, which is linked to the ribose sugar, and which is linked to the nitrogenous base. And now let's dive deep. So what are their classifications? Well, they can be classified into the two groups according to their nitrogenous bases. They are pyrimidines or purines. So purines are mainly adenine and guanine, and the pyrimidines are the cytosine, thymine, and uracil. And we will later on discuss when is each one of them used and how. And other than that, when we read the code, for example, of the nucleic acids, we use the A, G, C, T, and U letters for doing so. Now, let's continue about how the nucleotide sequences are being formed. Well, they follow the same dehydration reaction principle, which we mentioned numerous times in our videos. So what happens that when we want to synthesize new nucleic acid, we take the 3-phosphate variant of this nucleotide, add them together, and we attach this phosphate bond to the OH of the ribose, and we get the larger nucleic acid, and we also get the pyrophosphate, which could be also used for energy source later on. Well, let's talk about some structural traits of nucleic acids, since we know how they are being formed. Well, for example, let's take this variant. Well, for example, in DNA or RNA, which we'll talk about later on, the guanine can only form hydrogen bonds between the cytosine, and the adenine can form hydrogen bonds between the thymine or the uracil. Well, why is that? Because if we look at the picture, as you can see, the three guanine and cytosine can form 3 hydrogen bonds between them, and the adenine and thymine can actually form 2 hydrogen bonds between them. So why does this help us? Well, such bonding helps us because it is easier to replicate just such a double strand material, because one strand can specify what other strand looks like, so each strand is complementary to each other, because we always know that guanine is connected to the cytosine, so at the opposite side we should always have the cytosine, and for adenine at the opposite side we should always have the thymine. Now, another characteristic is that one strand has the 5' start, meaning that the phosphate is attached to the 5th carbon on the ribose sugar, and the strands also have the 3 start and the 3 end. This means that they are anti-parallel of each other because they have the different starts and different ends. So the 2 strands are complementary and the anti-parallel, and for example in DNA we always know that adenine and thymine should be into the equal quantities, and also the guanine and cytosine should be also in the equal quantities. Now let's discuss types of the nucleic acids. Well, there are 2 general types, which are the DNA and RNA. So DNA, the main structural difference is that DNA uses a deoxyribose and RNA just uses a ribose sugar. Deoxy meaning that DNA's sugar is missing 1 oxygen, and the DNA uses a thymine and RNA uses a uracil, which gives RNA much more flexibility. And also RNA is mostly found in the 1 strand, but it could be also found to the double strands, for example in viruses or other organisms. Well, let's talk about what their primary structure is. So the DNA is the hereditary material. As we talked about the mitosis and meiosis, this material gets duplicated into the S phase. So it is the very stable structure which contains genes and other things, which could specify events inside the cell. But the RNA has the numerous functions, but one of the functions is that it contains instructions about the protein synthesis. So it encodes the DNA's gene and later on delivers such instructions to a ribosome to transcribe the proteins. We'll talk about the transcription and translation later on into the videos. So, in conclusion, nucleic acids are the polymers made from nucleotide monomers such as adenine, guanine, thymine, cytosine, and uracil. They are divided into two groups, DNA and RNA, which differ in their sugar and structure and their primary function, and they are essential for storing the instructions for life and ensuring that the genetic material is passed on to the new cells. Thanks for watching and see you soon.